Next, this is a structure of mitochondria. As we said, it is going to be a human mitochondrial DNA. Before we have found the characteristics of mitochondrial DNA, it is going to be transferred through gene line, single gene line, family gene line. This mitochondrial DNA is going to be as circular, arranged in a circular chromosome, as double stranded manner, as human empty DNA characteristics, and the base pairs is also 16 base pair. So this is the structure of mitochondrial DNA. A human cell contains 100 to 1000 copies of mitochondrial DNA. It is the average copy, but it is going to be the copy number going to be different based on the mitochondrial size. So about 100 to 10,000 copies is going to be a human somatic cell may contain. As we said that 16 kilo base pairs. That's what we have mentioned here, about 17,000 base pairs to 15,000 base pairs, which means a kilo base pairs we have mentioned as 000. So a human mitochondrial DNA is going to be circular in manner, double stranded in manner, and going to be 16 kilo base pairs in size, in length. A major region of mitochondrial DNA is heavy strand and light strand. These two strands are going to be differ in their nucleotide content, as the heavy strand are going to have more nucleotides than the light strand. So the heavy strand, in the before structure we can see there is a heavy strand. This is a heavy strand. From here to here is going to be a heavy strand, but here is a light strand. So light strand is going to be found as a smaller nucleotide content, but the heavy strand is going to be present as the higher nucleotide content. So the heavy strand, as we said, the higher nucleotide content is present. It is going to consist of 28 genes, but the light strand is going to consist of 9 genes only. As we said, they are about more nucleotide content. It also consists of more amount of guanine residues, where the L strand, that is a light strand, is going to be rich in cytosine residues, while the heavy strand consists of more amount of guanine residues. The heavy strand is going to has a rRNA about 125. It can produce about 28 genes, which has more guanine residues, can produce about 125 rRNAs, which can be helpful in translation. That means more proteins can be produced through the heavy strand, and 152 mRNA, here I am mistake, mRNA, and 14 tRNA. That means it can produce about 12 polypeptides. Heavy strand can produce 12 polypeptides, but the light strand has only 9 genes, so it can produce 8 tRNA and 1 polypeptide protein. So as we said, it is going to produce a protein through this tRNA or RNA production through this 28 genes or 9 genes. What is the protein a mitochondrial DNA can produce is our question. So a mitochondrial DNA can produce its own proteins. A mitochondria is going to be a powerhouse of the cell. It is going to consist of a more amount of complexes. These complexes needs a protein. It is in general it was known as a protein complex. Mitochondria consists of five protein complexes. These protein complexes are helpful in biological metabolism. These protein complexes synthesis can be made in the mitochondrial DNA. It has a gene for this protein complex synthesis. So a human mitochondrial DNA is supercoiled to 37 genes, as we said before, and 9 genes and 28 genes. It is going to produce the OXPHOS, which means oxidative phosphorylation synthesis. Genes are responsible for the proteins complexes of oxidative phosphorylation synthesis proteins and mitochondrial protein synthesis. Mitochondrial protein means a membrane proteins. It includes the membrane proteins. In general, a mitochondrial DNA is going to consist of 13 subunits and 5 complexes. As we know, a 5 complexes what in OXPHOS, oxidative phosphorylation synthesis complexes, or the 5 complexes. They are NADH, ubiquinone and oxidoreductase. In general, we know known as NADH oxidoreductase, succinate oxidoreductase, cytochrome C oxidoreductase, cytochrome C oxidoreductase, and ATP synthase. All these are the complexes. For these complexes only, the mitochondrial DNA carrying genes information consists of. This mitochondrial DNA carries the information for the synthesis of these complexes. Here we can see they are about, this is the mitochondrial DNA of double stranded. Here we can see the L strand, H strand. Here we mentioned that 12 is RNA, 16 is RNA, which means these are RNA are synthesis from these genes. Here we can see 5 complexes as mentioned. A green regions are going to synthesis the complex 3. A violet region are going to synthesis the complex 5. A red region are going to synthesis complex 4. And complex 1 is blue region. Like that, a different 5 complexes can be synthesized from this mitochondrial DNA. As we said, these genes are going to be packed with a few non-coding, with only few non-coding nucleotides, because this is going to pack more amount of complex proteins information. So it consists of only few number of non-coding nucleotides. As we said, in the chromosome, which has a telomeres, but a mitochondrial DNA not has a telomere. A telomere is also a one type of intron. So a telomere is going to be absent in the case of mitochondria because it is found to be in a closed system. No information can be deleted off at the end of the DNA of a mitochondrial DNA, and also the replication to be followed out by the mitochondria is different from the replication carried out by the telomerase. Both are going to carry out the semi-conservative mode of replication, but there is a differences in their replication. So these genes of mitochondrial DNA are going to consist of only few non-coding nucleotides, but there is no introns. Introns means nucleotides are not even converted into RNA, but it consists of few non-coding nucleotides, which means it can be converted into rRNA or tRNA, but it not converted even to RNA. These nucleotides are not present in the mitochondrial DNA. A mitochondrial DNA is going to be similar in all these species. It differs only in the small amount. Only few differences are going to be found in the mitochondrial DNA, both within the interspecific and intraspecific. It is going to be very important characteristics of a mitochondria, as only few characteristics differs is going to be important characteristics which is very helpful in the DNA barcoding. Next, we are going to see about the replication of a mitochondrial DNA. It is going to follow the semi-conservative mode of replication, which means a parent DNA can produce a single daughter DNA during the replication. This is known as semi-conservative mode of replication. As similar to the DNA synthesis, it is going to use the DNA polymerase, but A specific DNA polymerase, that is mitochondrial DNA polymerase, is being used in this replication. This replication begins with the formation of displacement loop, that is D loop followed replication, which means there is a origin in the mitochondrial chromosome. Mitochondrial DNA is going to be looped off and the replication is going to be taken place. Next, as we said that mitochondrial DNA is going to have no introns. As it has no introns, between the two species, mitochondrial DNA has few differences in interspecies and intraspecies level. So, this main characteristics of mitochondrial DNA is going to be used in this DNA barcoding. So, the mitochondrial DNA has many genes. Among these genes, an important gene of cytochrome C oxidase 1 gene is going to be used as a standard gene. This is going to be a standard registered gene. It is going to be a reference to identify the species. So if we are going to identify a species, this cytochrome C oxidase gene is going to be a reference gene. Based on this reference gene, a species is going to be identified. So this reference gene of cytochrome oxidase 1 gene in mitochondrial DNA is going to has about 648 base pair length. It is going to be differ a small amount in each animal group. As the difference is small, it can be easy to identify the species. So a gene which differ only in a small amount is going to be taken as a reference to identify the different species. So this is a DNA barcoding process. If we are going to identify a certain species, a species is collected first. Then a DNA of that species is going to be extracted. Then the PCR amplification, which means a DNA is going to be amplified. That means a more amount of DNA can be available for us to identify. Then the sequence is going to be taken place, which means a nucleic acid sequence is identified through Maxon-Gilbert. Like that method, a sequencing is going to be taken place. Then the barcoding analysis taken place, which means a barcode of mitochondrial DNA, that is cytochrome C oxidase, is going to be compared with this DNA, with the sample DNA cytochrome C oxidase. When it was compared, a small difference is identified. Then the species in the sample can be identified. This is the DNA barcoding process. So in this video we can know about the mitochondrial DNA. Thank you.